Very pleased to be here. This is a really great event. I was also here last year, but as a guest, so it's really an honor to speak today. So my title is 5 years of long reads from research to clinical. Obviously, 5 years is not really correct because long-read sequencing has been around a little bit longer than that. I came in touch with it in 2014, actually, when we sequenced the first cancer genome on the RS2 BugBio machine. These machines were also named as Mini Coopers because that described best their size and the weight of the machine. Always kind of joke that there are kind of two really great achievements from the Genome Center, one of which is, of course, the contribution to genetics and clinical genetics in the human genome. Second of it is to install one of these Mini Coopers on the 15th floor of our building that needed some reinforcements. So it's been a wild ride. So 5 years mainly because before that it was really the dark ages. So like also in like 2015, the first mini ions from nanopore came around to be useful. I remember for many months, one of our computational guys actually held the lab record in running one of those machines. That's how unstable and unreliable they were. And then the Prometheans came around, which was a much larger scale from nanopore. And we did some test runs, and then all of a sudden we got an email, be advised, you can get electrocuted by this device without issuing any repairs or fixes. So these were kind of really the old dark ages from long reads. And I think since 2018-ish around, these technologies came around really significantly and are much more matured now. So that was one of the motivations why I chose 5 years. So as Todd said, I'm very motivated in understanding the mechanisms and the impact of structure variants in general in human diseases, but also in other crops and in other phenotypes. Just to define what are structure variants, these are like often defined as 50 base pairs or larger genomic alterations. You can differentiate them between deletions, duplications, insertions, inversions, and translocations. The really fascinating piece is like, although there are 4 to 5 million single nucleotide variants per human genome, whereas we only see like around 25,000 structure variants per human genome, so like 0.1% or something like that of events, they actually impact a larger number of base pairs on an individual, which makes them quite interesting to study. And so over the years, my group, but also of course many others, have mainly identified the roles of structure variants across these 4 areas, being in evolution, where we found multiple speciation events due to structure variants in our phylogenies. Of course, nowadays we are studying mostly genomic disorders. This is a diagram of a breast cancer cell line where you see all these crazy connections forming new molecules, which are due to structure variants, and that gives you more newer disease targets as well as newer molecules that you wouldn't have seen otherwise. And then impact on regulation, where we recently published a Cell paper which kind of nicely lay out the landscape here. And then of course, coming all of these together is this little plot here that I still like to show, where it has the heritability here on the y-axis and then certain selected traits on the x-axis. In red, we colored the copy number variations. In black, the rearrangements, and in gray, the single nucleotide variants. And so the motivation from this plot and from this publication is really once you combine all these variants together, because they're not just single nucleotide variants, but also structure variants and epigenetic changes in your genomes happening, then we get a much better view, a much better picture of what's actually going on in certain diseases, in certain phenotypes, and so on and so forth. So it's really important to discover these all. And as I said, like in the first slide, there's of course a longer history. It all looks like so easy when you stand here and present. But there is, of course, a long history. So like our understanding from structure variants really arose by karyotyping, where we can talk about gains of chromosome arms or chromosome arm losses into the genomic microarray area, where we can identify all of a sudden little genes or little areas of the genome being higher or lower presence than before. And then around when I started my PhD in 2008, 2007-ish, so Lexa and now Illumina came around, which gave us a much wider picture of our human genomes or genomes in general, right? Much more cost efficient, much more detailed. And then as you will hear me argue today, single molecule sequencing with the BugBio machine as well as with the nanopore machine really broadens the horizon much further to give us access into other regions. And so over the years, we really kind of utilized this technology and grew with the technology basically to have more insights into not just structure variants, where we can call many more structure variants with long reads than with short reads, mainly falling short, as I like to joke, about insertions and some complex regions, but also gives us much more details about repetitive regions in the human genome, many of which are actually medically relevant. So here we just published last year around 400 medical relevant genes, a catalog of them and a benchmark of them to be really important. And of course, as Todd mentioned, the T2T genomes is really then enabling us to study the impact of centromeres, but also of certain short chromosome arms that are now resolved. And as such, this was only also possible by utilizing long reads. And therefore, also we have an improvement in assembly and in phasing, where we can study the interconnection between different variants on the same molecule or not. So this is all what long reads brings to the table, so to speak. And there are some publications from my group, but there are many other publications that you can read up if you are really interested in more details. So with all these advancements, we were really eager, like a year or two ago, to kind of put them to the test and put them to the action, right? What can we really do with all of these new things in terms of human health? So together with Stanford, but also Google Health and UCSC, we set this kind of ambitious goal. We want to create a study where we are open for patient enrollment in the Stanford clinic. And what we really want to do is like really go from A to C, from blood draw to clinical report or clinical research report in the fastest time possible, right? So give you a perspective, clinical sequencing these days is still kind of rough, right? So usually it takes like a month or two before you get your test results back. And the usual solving rate is around 15 to 20%, not much more than that, right? This has multiple reasons that I don't want to get into it, maybe all the panel discussions. But basically what we did over a set of 13 patients that we could enroll over this year, we had a solve rate from 46%, and we went from blood to clinical report. Fastest time was 7 hours and 18 minutes instead of 2 months, right? And that really opens up the gate, because like then we can turn around critical care patients within the work shift of a nurse, right? And give the doctor at least some genetic hints if there are some. We're not claiming that everything is perfect here. And this was in part only possible to do multiple innovations, not just on the sequencing side, but of course also on the engineering side together with Google and their teams to fully leverage the cloud infrastructure. So just this slide is just to show you that we didn't cherry pick our patients actually. There was a wide range of years of age, so from 3 months to like 57 years. And also different phenotypes, lots of it is kind of cardio arrests. But we didn't kind of limit ourselves to those individuals. And I want to walk you through this kind of study a little bit and how we did this with the case of FAST-002, which is actually a 13-year-old boy called Matthew. So this was back in 2001, 2022. Matthew developed kind of some 2 weeks history of dry cough, decreased appetite, chest pain, and severe fatigue. And of course, everyone thinks, well, this is COVID, right? Like, that was exactly the time where we had to deal with it, still would deal with it, but the symptoms fit perfectly. So he admitted himself, or his mom admitted him, to the hospital in Oregon with the suspicion of COVID. But what actually was going on was like he had a heart failure actually. And so based on the diagnosis, they airlifted him to the Children's Hospital in Stanford. On the arrival, he actually went through a cardiovascular shock and had to put on the heart-lung machine. And now the question arose, like, what should we do, right? It could be either a genetic problem. If it's a genetic problem, we can transplant its heart, and this boy is going to live happily ever after, so to speak. Or if it's an environmental problem, then like the heart transplantation wouldn't help, right? So this is kind of the really tricky question that we were facing. And so he was enrolled in our program, but simultaneously, of course, they also enrolled him into standard clinical sequencing from exon sequencing. So what did we do? Our PhD student took the 2 milliliter of blood vial and ran across the campus to start the DNA extraction, the DNA fragmentation, and the library preparation. So note here, this is around the 222 kb fragments extractions. And he did this all in 4.2 hours in our lab in Stanford, or in the lab in Stanford. And now one of the, and this is of course also already optimized, but it wasn't the fastest run. And now the kind of trick starts. So we used the Promethean machine from Oxford Nanopore that has 48 cells where you can sequence the human genome in. So the trick here is actually we loaded Matthew's DNA in all 48 flow cells simultaneously and pointed the sequencer directly at the cloud. So within 162 minutes, we actually accomplished 63x human genome sample, right? This is like blazing fast. They actually needed to update their Infinite connection. And then simultaneously to the sequencing, the base calling already started. With the alignment, you can see the chemistry was still a little bit older one. So like we have an error rate of around 95%-ish these days. It went a little bit better to like 98%. And the variant calling started. We were mainly focusing on single nucleotide variants. Just a few structural variants from my side were included. This was mainly because of interpretation. It's still hard to be certain what the impact of the structural variants are in a clinical setting. This is just due to we need more curations and need more references. So this took all 3.6 hours, but was overlapping heavily with the 2.7 hours of sequencing. After that, we took the 4 to 5 million variants on the computational side, characterized them, did population frequency thresholds, and Omen morbid scores. So we went down to the prioritization after the computational prioritization to 18 variants, with 10 of which had a higher morbid score than 4. And then the curation team went in there, which also took a little bit of a few hours, and they kind of really are an amazing team. Those are like 3 women that really study all the variants and have to make a decision if this variant that they actually see is believable, first of all, but also does it fit the history of the patient. And so what they determined was actually that this little purple site here is a duplication of this T site, which is actually a genetic cause of this heart failure. So we immediately could enroll him for the transplantation. And in the short story is he got a transplant, and he's living happily ever after. We actually got a Guinness World Record of sequencing the fastest human genome for this work. We actually celebrated it with Matthew. We invited him back to Stanford for that. And so you can see Matthew was actually down here, one of the first cases that we had, not the fastest. But then here in the second set of flow cells, we optimized really a lot more. And you see how the run times shape down, the different colors or the different stages in the experiment. We published a study in New England Journal of Medicine as well as Nature Biotech for the computational aspects. So this is all great and wonderful, right? Right in the first 15 minutes or so, I'll show you a concrete example how long reads can really shape the future in clinical sequencing. But of course, I have like 45 minutes, 30 minutes left. So like there's a little bit more than that. And so of course, we ask ourselves, how can we improve things, right? This is great. There's actually now a second phase just started where we broke our old record beneath 7 hours turnaround time. But how can we improve things? Well, one thing that we wanted to improve, and which is also more of the focus on the second phase of this project, is structure variant calling. And so for that, but also other reasons, we implemented a new version of our structure variant caller, which is called Sniffles version 2. And here I'm just showing you some benchmarks for completeness. So up here is a genome-wide benchmark. Down here is the challenging medical relevant gene benchmark from Genome in a Bottle, which is a NIST effort from the US. And basically, you can see we're doing great. So these plots here show the accuracy of the genotype F1 score on the y-axis, the different programs on the x-axis. In blue, you see the default parameters from the individual programs for different coverages, which are the tick marks. In red, you see that we tried to tune the parameters to improve the results from the individual programs. The transparent tick marks down here on the bottom are just runs that perform below 50% F1 score. And you can see here nanopore data on the top, genome-wide, and nanopore data here on challenging medical relevant genes. Sniffles 2 is actually doing exceptionally well, outperforming the others pretty well, even with like 5x coverage or 10x coverage, which is the star and the square. Here's also just for completeness the hi-fi data set. Same results, basically, we are outperforming the others. And what was also important for us is actually going after the runtime. So kind of the spirit of what I just showed you. Here you see that Sniffles actually outperforms the other ones as well. So this is great. We are more accurate and faster, like every other method that is getting introduced over such seminars. But like, there's much more I want to tell you about this and much more trickiness that we wanted to kind of solve. So one of the things that I was really motivated was that so far the community have mainly looked at 1 or 2 or 3 or 4 genomes at once, right? But there's a clear trend going on. So this is a figure from our review paper where you can see on the x-axis the time of years up when the paper was published, and then sample size and the different marks of the different genomes. And so what you can see from this is that there's a clear trend of like more and more long-read genomes per study, even for larger genomes, right? And so this trend clearly continues these days with all of us, where we are sequencing 10,000 long-read genomes, but also with like other things that I'm going to highlight a little bit later. So we need a method that doesn't just work on a single sample, but actually scales to populations. And so what we did here is to come around this kind of old problem where we have so like the traditional approach right now is to do SV calling on a single sample, merge the results across the samples, but then we need to identify if the variant in sample A is really present or absent in sample B, which is not so easy because you can be tricked from your caller because of certain thresholds or certain coverage fluctuations or whatever. So you have to read genotype, go back to the BEM file, and then merge these results, and then you have a population fully genotyped VCF file, which is really your go-to place for any GWAS study, for any comparative genomic study. So in Sniffles 2, what we did, instead of just putting out the VCF file, we actually also put out a little binary file here, which is just like a 2-digit file size per sample. And this little binary file holds any candidate that we ever observed in this in particular sample. And so what you care about is the VCF file if you're just interested in this single sample one. But the binary file we use for merging, and so we can go back and say like, well, is there any evidence of this variant in sample A, given that we're looking in sample B, right? And so this speeds up this whole process significantly. So instead of 36 hours, we just need 65 seconds for one family to produce the same results, and actually even a little bit better. The cool thing is it solves the n+1 problem. So if someone wants to bring in, I don't know, a cousin or some other samples to this study, we can just add this onto it, and it doesn't take much longer. For this approach, we would have to start at this point again and go through everything again. So it really scales nicely population. It also improves to more normal comparison for cancer patients, for example. So the first thing we did, we looked at Mendelian consistency, which basically is a measure of how well does the caller fit the inheritance pattern within the family. So in blue, you see the proportion of variants that fit the Mendelian consistency. In red, you see the ones that don't fit the Mendelian consistency. And in yellow, you see the missing genotypes. When we compare this now to a single sample calling without re-genotyping, you see it's a lot of genotypes are missing. There's a lot of uncertainty. And then when we do the genotyping, which is in the 36 hours, you see the consistency is higher, not as high as our approach. And there's still some missingness and a little bit higher inconsistency. We actually put this a little bit more to the test. So we kind of merged 770 genomes with Oxford Nanopore. And this also took us just 15 hours instead of 36 hours on a single family with the traditional approach. So I would argue this is really kind of boosting and opening the door for family studies, population studies, to monomer studies in larger settings. And so we did this on the MACB2, on a cohort, a Mendelian cohort around MACB2. MACB2 is a very interesting gene just on the end of this chromosome X here. It's a very tricky region. So we sequenced around 30 or so samples, and 95% Sniffles could identify not only the correct location of the de novo allele, of the causative allele, but also correct allele of it. There was one case where we couldn't do this, and I'm showing you in a second what this is. The MACB2 is actually a profound neurological and developmental delay that is affected in male, and Claudia is really the PI behind this study and helped us a lot to understand and make sense of it. So this is the one case where Sniffles didn't really recover the entire allele. This is still a challenge for us right now. So what you see here, so Here's, where's MACB2? MACB2 should be somewhere around here. There's MACB2, there's the MACB2 gene. There are three segmental applications highlighted in orange here. And in blue is the call that Sniffles made. So Sniffles was able to identify the de novo allele, or the de novo variant, just didn't recapitulate the entire allele. And the reason for this is the following. So based on array data, but also other experiments, we know that in this region we have a duplication in this area, a triplication in this area, and a duplication in this area. Actually, what we also know from other experiments is that this triplication is also kind of inverted. So the actual allele that this patient has is highlighted down here based on these error signals. And so Sniffles currently cannot resolve this entire complexity of an allele. It's actually really challenging to report this within the FAR standards right now for the outputs. But it's nevertheless something we are going to continue to look into, that we can resolve these things much better in future. Overall, it was actually a really good result. We identified almost all the alleles that were causative, and most of them we could resolve correctly. Here's a case of a tumor normal comparison of the call 8 to 9. This is just a standard cell line that many people are studying. We can identify within seconds. Here's the normal tissue, here's the tumor tissue. Sniffles can identify these deletions within seconds, causing 2 exons of P10 to drop out in the cancer tissue, which is a really important gene for the development of cancer and so on and so forth. So this is now very easy and done within seconds. So we are applying this, one of which is Gregor, which is this Mendelian cohort, part of which I just showed you a little bit over the MACB2 to solve the unsolved Mendelian diseases right now. This is a project that is continuing, ongoing. In the All of Us research, we are also applying Sniffles 2. All of Us is a really, really cool program actually here in the US. If you ever want to get your genome sequenced for free, you just have to register. You get all the data back if you want to get also a report for this. So like if you ever want your genome to be sequenced, this is the perfect opportunity, and it's for free actually. So they are going to sequence 1 million Illumina, they were sequencing 1 million patients on Illumina and 2 million patients over array analysis, and report the findings directly back to the participants. So you don't have a doctor in between, which is kind of a very interesting experimental model. We are all very curious how it plays out. Within that realm, the HGC in the first year sequenced around 1,000, 1,300 long-read genomes, and we are going to ramp this up in the next year of additional 2,000 to 3,000 long-read genomes that contribute to this across participants that are being chosen randomly, but like with some preferences on some phenotypes. And then we're also working together with the Emirates. G42 just renamed themselves to M42. And they have so far sequenced already 85,000 whole genomes with Oxford Nanopore, which is a massive endeavor. They're investing this because they want to create an annotation database for the population. So this is all great and fun. So I showed you it works better on single sample. I showed you we innovated on population scale to enable this, and Mendelian studies a little bit further. There's still so much to be done. The other aspect that I wanted to kind of dive with you into a little bit right now is somatic or mosaic structure variants. So as you know, we have germline mutations that occur very early on, and then they're getting spread out through your body. But somatic mutations or mosaic mutations are happening during your lifetime, basically, and are usually confined to certain locations in your body. They are low-frequency mutations, sometimes really hard to identify. The worst form of this is, of course, cancer mutations, but most of the time they are more or less harmless. So the challenge here is, of course, the signal-to-noise ratio, right? Because like we have, for example, a variant like this, where we have like 3 insertions in 80x coverage data, 84x coverage data. And we want to identify this variant, and short story is we can identify this variant, but we also don't want to kind of open the floodgates for all these false positive variants that we potentially also identify if we just find everything. So to account for this, we first, of course, started with kind of a spike-in experiment. We are spiking in HG002 that we know so much about into HG733, where there are high-quality assemblies available. And after the spike in of HG002 at different concentrations, 7x, 10x, 14x, 21x, and 28x, we asked back how many variants we can identify and how many additional variants outside of HG002 were identified. And so as default setting in blue, but also other SV callers, this is more or less what you're getting. So here's your precision, which is pretty low because like most of the variants that are identified is HG733 and not HG002, so a different sample. And then recall is also pretty low because like they were not meant to call down to like 7 or 10%. When we now enable our mosaic mode in Sniffles, then the precision is much better because we are reporting those allele frequencies where HG002 is happening. However, we saw that our recall is still a little bit iffy. So especially here, this is very demotivating or very frustrating actually, right? And so it took us a while actually to figure out what is happening here. So what is happening in the spike-in experiment is we're actually sampling the reads that carry variants, right? We are sampling them at 7% rate. But actually, most of the variants are heterozygous variants, right? They're not occurring in all the reads of HG002. So by sampling them, they're actually present in just 3.5% of the reads after the spike in, right? And so when we account for this shift and saying we only want to benchmark variants that are actually present in the data set, then our recall goes up, and this is the green bar. So it's a little bit cheating because we are correcting for what we actually observe in the data, but I think it's fair enough. And so that's why I'm always showing both results. So in the allele frequency spectrum that Sniffles was designed for, 5 to 20% of all the reads, the green bar is the recall rate, which is much better than default or other SV callers. So this is great. This is spike in, a little bit boring. This is just for kind of sanity's sake. What can we actually do with this? And so this is one of the figures that are still one of my favorite figures since like 3 months now. My lab mates are growing boring of this figure, but I think it's amazing. So let me walk you through. So we took a brain sample, sequenced it with 55X in the center. It's actually an MSA, so a neurodevelopment disease sample, a part of a brain from a patient. Again, we are hunting at this kind of neurodegenerative disorder. But what we actually found is not related to the disease, but we can find something really cool. So what I'm showing here is an IGV screenshot. On the top is nanopore data, on the bottom is Illumina data. As you can see, here is like a really big band of purple. This represents a homozygous insertion of an ALU-Y element, actually, so a repeat element of 3,400 base pairs. What I don't show you here is actually that there's also an ALU element here in the genome already. And so what Sniffles can now identify is not just this ALU-Y insertion, but these little black lines down here, it recognizes that this is a deletion, which is correct, of a low frequency. So this is a mosaic event of like 5–6%, which actually represents that this ALU element here gets recombined with this ALU element here, right? And so this is really cool. Trust me, it's really cool. Here you have the validation gel. So you see the big band here for the ALU insertion, and this little smear here, which is the deletion here. You see here the control brain doesn't have any of those two alleles. This is really cool because now we have a tool that can operate on like 1 or 2 runs of Oxford Nanopore to detect these events. And so in the brain itself, we can identify 26 of exactly these kind of events that have so far been often overseen by biologists or geneticists. In total, we found 125 insertions that lead to mosaic deletions. We think it's kind of an instability of the region where we see the insertions itself, right? So this is really, really cool. That's why I like this figure so much. I'm going to come to another figure that I like a lot, though. So the other thing we are doing, so this was one brain sample, right? But to kind of promote this population mode together with what I just showed you, this mosaic mode, what we are collaborating with is CARD, which is an NIH effort, where we have sequenced 4,000 brains, a few of them really deeply, to understand better this impact of these mosaic mutations. So the one brain that I'm showing you here is actually really cool. It's a complete control brain, so no disease, no nothing. But you see here we sequenced 4 different parts of this control brain. The frontal cortex with 400x coverage, the cerebellum with 800x coverage, the other sides with 70 and 60x coverage. All of these have 30x BAC biogenomes, some short-read RNA-seq and some single-cell RNA-seq. And this is really, really nice, and actually happened all by accident because this control brain was simply used for training the technicians, and the technicians never stopped sequencing. So this is not great for us to have this as a resource and to study this. So we were wondering how many mosaic structure variants, so the thing that I just showed you, low frequency mutations, are happening per region of here. No one has ever looked at this, honestly. The only thing that people have looked into is low frequency single nucleotide variant mutations in the different parts of the brains. And so, let me see if I don't mess this up. Yes, the frontal cortex was shown in the SNP study to have higher rates of mosaic mutations than the other regions. And so when we do the same for structure variants, we see here the different regions, so cerebellum, frontal cortex, and so on and so forth. We have different events that sniffers can detect, and these are all potential mosaic mutations. We are still going through and validate them and filtering them and so forth and so forth. But the cool thing is we see the same pattern that the frontal cortex has a higher number of mosaic mutations than the other regions in the same brain, which is really interesting. And the consequences of which is unknown, but we are trying to get around of this. So you can see here some examples where sniffles actually did a really good job. So for example, here it detected that there is a mosaic deletion here, which wasn't detected in other tissues. In some scenarios, we didn't do a good job. So here, for example, we thought there is only this insertion present in one sample in purple, but clearly it's present in the other three tissues as well. So this is certainly something that we still need to improve a little bit, maybe also potentially with the help of AI at some point. But that's still a little bit work in progress and still a little bit challenges. So here I'm showing you another gel, which looks really ugly, but these regions are mainly in repetitive regions. So it's a little bit tricky. So let me walk you through. So this is a deletion that we predicted actually to be around 6% and unique in one of these brain tissues. You have to trust me. The deletion actually validated in this gel, but in all 4 regions, meaning like we didn't get it right to be saying that it's just in one tissue. We still have to figure out if it's a sampling issue from the sequencing or something else going on. The cool thing though is like we also had blood from that patient, from that individual, and there we don't see this structure variant, this deletion occurring. So it might be a mosaic mutation, not just in one region of the brain, but spread throughout the brain, which is not inside in the blood DNA, or potentially somewhere else in your body. Really cool, right? That's nice. So it's my second most favorite one because we didn't get this thing right. Okay, so where we are applying this, I already said CART is a big thing where we are applying this. Of course, cancer projects, the Canadians, as well as Genomics England, we are working together with them to improve the cancer detection pipelines for both of their patient groups together with sniffles. And it's really working out nicely so far. There's a nice paper coming soon together with the Canadians. One little aftertaste that this still has, though, is the following issue, right? So like we have bulk nanopore sequencing. Our detection rate depends really on the coverage or the variant allele frequency that this mutation is inside of the sample, right? Which is a little bit tricky. However, we also have single-cell short-read DNA sequencing, where we then get plots out like on the right side here, where we see certain duplications marked in red, or certain deletions if you see any in blue here. But the problem is like the resolution of these events is horrible. We see like 500 kb events with plus minus 5 kb. This could be including a gene or excluding a gene. Who knows what the hell is going on? And it's really hard to prepare and everything. So the question is like, can we combine this like single cell assay with the resolution of the long read sequencing? And in short, this is what we are doing. So here I'm showing you some examples. So this is one region of the brain. Actually, this is one single cell of a brain sequenced with Oxford Nanopore. On very early data, we have now collected many more data sets. So all the reads are coming actually from a single cell. And I would argue it's one of the very first times we actually see these kind of larger insertions or allo insertions or repeat insertions, repeat expansions in single cell data per se. And this is only with the help of these new technologies, of the long read technologies. So this is something really, really new in collaboration with Christos. And we are working our way through it. improving the analysis strategy, and of course, lab methods. So this has been already a good marathon. So I have one more chapter to kind of put you to sleep in the beginning of the conference, which I hope is not the case. So far we have talked about how long-read sequencing can really enhance our ability not just to diagnose, but to speed up potentially the diagnosis of individuals, right? I've shown you our work on structure variants, how we can improve this, how we can improve population scale, comparison, as well as mosaicism. One of the things that I haven't talked about, well, there are many things I haven't talked about, but one of the things I haven't talked about are tandem repeats, which are really, really interesting. They are really high repetitive regions in your genome. They're more prevalent than you maybe think. So like 8% or something like that, 8-9% of your genome are tandem repeats. And actually 70% of all the structure variants occur in tandem repeats, but there are also a lot of single nucleotide variants in these tandem repeats. So it's really important to get them more to know, basically, because they are also already reported to have impact on around 60 diseases, Alzheimer's, for example. But they're really, really tricky. And of course, they also are the source of mosaic and epigenetic changes that we're currently trying to study. So how are we doing this? So the following is mainly BugBio data, to be honest, and also a tool that we developed together with BugBio, which is called Target, TRGT, which is the tandem repeat genotyping tool. And what we developed here is basically not just a caller that takes in the long reads from BugBio, and also a repeat catalog, which is kind of normal. Here our repeat catalog was defined actually by us, being around 1.2 million regions of the human genome. But we also implemented a visualization tool for this. So here you see in blue is actually the repeat region. So here, for example, CGG repeating 30 plus times. And then in greenish, you see the flanking regions, because often it's also interesting to see what variations fit in the are happening in the flanking regions. And I'm going to walk you through this a little bit. So this is just what I said. So here you can see the visualization. We see like little insertions. This is how a little deletion looks like. And this is how a little mismatch looks like. So together with Target, we are not just aiming for genomic variants, but also to visualize and identify methylation changes. So here in red means high methylated, and blue means low methylated, right? We can obtain this also from the long reads. In the same sequencing assay, we can pull out the genomic variants, we can pull out the epigenetic variants, which makes this a really powerful tool and technology to get a comprehensive overview of your genome. So to assess this also, we put together a little benchmark of how good this tool actually works. So just in short, without further ado, so based on an assembly comparison, we have a 97.70% agreement. And then like based on Mendelian consistency within a family, this is again HG002, 3, and 4. We have 89% concordance across all repeats, and 99% concordance if you are just allowing for one base pair difference across the family and itself. So this is pretty good. Adam English and I are also working on a benchmark, which I put in the slide in the very end of it. So the one thing I want to talk you through, because it's beautiful biology actually, and I hope I'm not screwing this up in the end, is the FMR1 locus. FMR1 is like a really, really hard to assess locus. It's on chromosome X again. And what makes it so tough is the repeats. So a normal carrier, so most of us in the room, or almost certainly all of us in the room, have around 30 repeats, 30 repeats in FMR1. There are people that have Fragile X syndrome, so pre-mutation carriers have 55 to 200. They might not have the phenotype of Fragile X syndrome right now, but they might probably develop it over the time of their life. And then a full mutation carrier that has a Fragile X syndrome has around 200 repeats. The tricky part is like with 200 times the repeat of the, I think, 4 or 5 character motif that it is, means that short reads cannot resolve it. Usually short reads end up around 200 base pairs or something like that. This FMR1 is kind of exploding this range by multiple folds, right? So it's really hard to characterize this. And so together with Penguin, David Nason, and Emily Allen, and our center, we have sequenced actually multiple brains of pre-mutations and full mutation carriers to get more insights into this Fragile X syndrome disease and others. But let's start easy, more or less easy. So this is a control sample, HG003. It's actually the dad of the one sample that I keep showing you all the time. And remember, it's on chromosome X, so the males are just carrying one copy, right? So in blue, we see it has, let's start slowly. So we see it has 28 copies, so it's perfectly in the normal range of what we think it is. And then in blue, you see the methylation pattern, which is low methylated. So based on literature, we know that only full mutation carriers have a higher elevation of epigenetic, of methylations. And so when we look at this whole sample in detail, we see most of the reads show a low methylation signal here, which perfectly fits what we expect. And then we have a sequencing data sets of 100 bio-sequenced males. No, not 100, but like a total 100 controls, some of which are males. And we see all of them are agreeing low methylation in this range. So this is great. How does it look in females? Well, there is a little bit more complicated. We have 2 copies of X, right? That's the easy story. We have one haplotype here and one haplotype there. Has a little bit of difference, so 27, 28 repeats versus 30 repeats. Now the tricky part, and I had to read this up again, is what is happening with the methylation here, right? We see a lot of blue, we see a lot of red, but in both haplotypes. Well, actually it is the case that exon deactivation in females follows a random pattern throughout the cells. And so it's not so surprising to see that the methylation doesn't follow the haplotype assignment. In autosomes, it would actually follow them, but not in the X chromosome. So when we look overall, female has a bimodular distribution of methylation, and basically all our control females have that same pattern. So everything is expected, and we see already some nice biology coming through, and I had actually to learn something, which is also great. Over all our control samples, we see most of them follow actually expected around 30 copies of this repeat motif in FMR1. Funny enough, we have 2 individuals in this control set, which are 1,000 Genomes project, that seem to have pre-mutation carrier points here. And that's a whole discussion if we should follow this up or not. And so here we are showing you some of our brain samples. Now I can dive into these brain samples that we have for full mutation, pre-mutation carriers, right? So on the top, you see 67 repeats and 123 motifs. So these are pre-mutation carriers, 1 female, 1 male. And sample 7 is a full mutation carrier, 366 motif repeats, right? Pretty cool. You wouldn't get this with Illumina sequencing at all, I think. And here you see also the methylation. Now is the time I sometimes screw up. So remember, we have a male. Sample 2 is a male here. It has a pre-mutation carrier. We are not expecting to see a rise of methylation for these pre-mutation carriers. So what we see here is a pretty low methylation rate across these repeats in that sample based on our tool. Then we have a female, which is a pre-mutation carrier. Remember, it's a pre-mutation carrier, which means we expect a low methylation, but simultaneous to that, the exon deactivation is also happening, which silences the copy of X over high methylation. So what we see is overall medium level of methylation in orange. And then here we have a male, which is a full mutation carrier, which based on arrays, we know that it should also come with a high methylation rate. And this is exactly what we see here in green. It's actually chopped up here, but you see this very nicely, right? So this is really, I think, really nice biology, and we are recapitulating what we are expecting to see, but this is really encouraging, and we are currently analyzing the whole genome data set, of course, of these free samples, and we have like much more samples already done on the BugBio machine. And this is just a shout out to Adam English's work about tandem repeat benchmarking, where we designed a new benchmark of 1.7 million repeats with a ton of new mutations in indels and structure variants. And this benchmark is cool because it's the first time it combines 5 base pairs and larger variants into the structure variant realm. And it can also deal with different variant representation, allele representations across tools and technologies. So it's pretty cool. We expect to submit this as a preprint around Halloween. We thought this is a fitting time for kind of spooking everyone with tandem repeats. That's cool. So summarizing, I hope I'm still in time. I'm good. Utility of long reads. I think it's pretty cool. It's pretty epic. So I think buckle up. It's coming. I talked a little bit about the speed of diagnosis. As I said, we are currently below 7 hours from blood take to kind of clinical research report. There are still lots of things we want to optimize, not just like cloud infrastructure, but also optimize machine learning approaches to further filter and prioritize variants, further to make the work from the curation team easier. Then I showed you our work in structured variant calling with sniffles. We not just improve single sample calling, but also make it easier to compare across samples from a family up to like 80, 90,000 individuals, which still requires some fingers crossing that it runs through on a decent machine. And then last, I showed you some more insights into the tandem repeats with target sequence resolved variants down to mosaicism levels and methylation assessments. And I also teased you one slide with a tandem repeat benchmark that we're bringing out. And I think what combines all these three projects together is really nicely this kind of notation that we want to leverage this sequencing technologies to full extent and really go into the regions of the human genome that have little to none been explored so far. And it's really challenging to kind of identify what these variants are doing that we are finding. But it's such an important first step to find these variants in the first step, right? Which we hope really drives forward not just biology, but at some point also kind of medical and clinical diagnosis. And so with that, I want to thank my entire group, bunch of crazy guys. Want to thank HGSC, Stanford, UCL, and PNRI for great collaborations and on the right side funding. And thanks again for inviting me, and thank you for giving me the opportunity.
Questions?
Everyone wants a break.
Thank you for the great talk. I have a question regarding Alzheimer's disease cohort, specifically the control group. What is the characteristics, if you know about this group, like the age, they have...
you mean the CARD project, or what do you mean?
Yeah. You showed that there are, in the CARD projects, there are 2 cohort patients and control.
Oh, you mean in the you mean these guys here?
Yeah.
Oh, so these are just like 1,000 genome samples. They're randomly distributed through ethnicity and population. And these guys, I don't know. Honestly, I wouldn't be shocked if those are all white brains, because like it's very hard to get African American brains.
I see.
Just from the biobanks.
Are you planning to use all of us data, which have, I think, different ethnicity in the same project, or it will be late?
Actually, not really. It could be interesting. I haven't thought about this. I think these 100 data sets here is giving us already a good control, because we can assess which tandem repeat is outside of the norm. The different ethnicities might mix it up with a little thing, but we could mix up the Hispanics and the African Americans in the all of us data set and use that as a control as well. Just not sure yet if I want to do that.
Okay, thank you.
Any other questions? I have a couple. We're doing well in time. Okay. So I guess just I'll stand by the mic so everyone can hear. So Fritz, so for the speed, yeah, I got a couple for you. So for the speed thing, it's really fascinating. So if, let's assume everyone's genome is sequenced and available, right? So when you go into those decisions, that you can just look up their genome. When would speed still be relevant? Let's say all of us sequences everyone's genome in the U.S., and then the doctors just have that available. So when would the speed criteria be? I guess you hinted at this, like things can change over time. I guess.
So I think that would be my first escape route to tell you that like genomes are not stable. They can change over time, as you can think like of UV or whatever environmental factors can play into this. Also, of course, with the mosaicism or somatic mutations, it's pretty tricky because you can have different mutations occurring in different tissues of your body. And so we just got funded over the SMAT grant, SMAHT, which actually aims at sequencing 150 individuals and I think like 8 or 10 tissues per individual to kind of identify how many mutations we expect in a control population across the different tissues. It's actually really fascinating because there's also a nice paper showing that everything that has a higher prevalence than 5 or 7% for SNVs are likely to be shared across all the tissues. For SVs, no one has done that, and the argument here would be that transposons wouldn't follow this. Because when you have early on set a mutation, how it kind of triples down through your cell lineage, that's kind of the motivation that this 5-7% comes into. But transposons wouldn't follow this pattern necessarily. The one thing that is pretty cool is that the one figure that I showed you, this one, with this PCR, we actually took this PCR and put it through different brains that we had available in the fridge, so to speak. Oh, no, that sounds awful. But the cool thing was that this allo insertion at this location is actually common in the population. And then what we found is actually that therefore this mosaic deletion here is actually also common in the population. So again, I'm not claiming anything disease relevant to here because we don't understand this mechanism, and we see also some of these occurring in interesting genes pertaining to the phenotype. But for me, it's fascinating that an event that occurs maybe just 4, 5, 6% in this one brain tissue is actually quite common in the population, which is due to the genomic instability between this allo insertion that is common and this allo element that is here in the reference anyways. And I think it's really, really cool, and we hope to see many more biology now happening with a tool at hand that can now detect these events automatically.
Yeah, yeah, that's great. I think another fascinating aspect or anything, and obviously, I focus on microbes, but anything that's low frequency but highly prevalent is always interesting, right? That's it.
Yeah.
So I guess one last question here. We have maybe one more minute. It's more of a kind of technical question, which is it's fascinating, the speed improvement you got with sniffles too. And you mentioned the N+1 problem. I guess just maybe briefly comment. So the idea is that you're kind of indexing them so you can do quick. So the problem is current tools would have to search over and over again across all the samples.
With the current tools, once you have completed this whole thing and you have a population fully genotyped VCF file, you add one more sample, you do the SV calling on this one more sample, but then you have to merge with all the other samples, and then you have to re-genotype across all the samples again, right? So it kind of is an exponential growth of compute and things that you have to do to get an N+1 done in this side here, right? And so by having these binary files available, you don't have to go back to the BEM files, which are the really bulky files and really the slow part of this whole thing. We know all these candidates in these binary files, given, of course, that we didn't screw up. And so we can just go through this, 50 MB or whatever binary files during the merge process and be done with it. So adding one more sample to the whole process is just taking maybe, instead of 65 seconds, 70 seconds. Instead, this thing here probably goes to 50 hours easily.
Yeah, that makes sense. I just was not quite clear. You're not recomputing anything. You're just...
We are not going back to the BEM files. Yeah, we have our temporary files, right, where all the information is in there that we actually need, compressed, and we can just work from them.
Okay, I guess the only challenge you would have is if you discovered, you mentioned one structural variant that no one could call, and then you make an improvement, and then you'd have to go back and redo it.
You would have to go back, yeah. Well, yeah, you will have to go back. There are certain scenarios where you have to go back, yeah.
Very good. All right. If there's no more questions, I think we're going to transition to a break, so let's thank again our speaker.
Thank you.